Cellular Endomembranes and Protein Transport, Part 4. So here are all the trafficking pathways that we were talking about in eukaryotes. So we talked about the vesicle trafficking from the ER to the Golgi. We then talked about a pathway where the lysosomal hydrolases are moved to the Golgi apparatus, to the late endosome, or to a lysosome. We looked at several pathways from Golgi to the plasma membrane, the constitutive secretion pathway and the regulated secretion pathway. Finally, we looked at a couple of endocytic pathway, which included the fluid phase and the receptor-mediated endocytosis pathway, which follows through the early endosome into the late endosome and to the lysosome. We also looked at a second endocytic pathway, that is phagocytosis. So you should be familiar with all these vesicle trafficking pathway. So in studying these pathway, some important questions should have come up in your mind, and here are some of these. You might be wondering that what is the mechanism by which the vesicles are formed, what actually provides the mechanical strength to bend the membrane and pinch the membrane off. You might be curious about what confers the specificity of the system. So you will remember that I said at the beginning that the process is very specific. When a vesicle buds from ER, it must fuse with the Golgi apparatus. It must not fuse randomly with other membranes of other organelles. So what confers this specificity? What makes sure that the vesicles bind to the correct target membrane? Finally, once that vesicle reaches the target membrane, what proteins regulate the fusion event, or how is that fusion event mediated? So that's what we will talk about in this last part, essentially the molecular mechanism regulating vesicle trafficking throughout all these pathways. So let's start with the mechanism by which the vesicles are formed. When we are talking about these different pathways, we encountered two pathways that depend on or used clathrin-coated vesicles. So these are vesicles that are coated with mostly clathrin, but other proteins as well. You will remember that one of those pathways was the pathway from the trans-Golgi network to the lysosome. So when vesicles bud from the trans-Golgi network, they bud as clathrin-coated buds. They pinch off the clathrin-coated vesicles. The other pathway that depends on the clathrin-coated vesicle is receptor-mediated endocytosis. But there are other kinds of coated vesicle in this system, and they are collectively known as coatomer-coated vesicles. There are two groups of them, the COP1 and the coated vesicles, and the COP2 coated vesicles. So these are called the coatomer-coated vesicles. We'll look at their functions in the formation of vesicles and how they function in this pathway. So here are three types of coated vesicles involved in the vesicular transport. We just mentioned this one. The clathrin-coated vesicles are in the blue, and here you can see the clathrin-coated bud forming from the trans-Golgi network. There's a clathrin-coated vesicle en route to the late endosome and to the lysosome. And likewise, here is a clathrin-coated vesicle forming at the plasma membrane. This would be fluid phase endocytosis or receptor-mediated endocytosis. The COP2, on the other hand, regulates vesicle trafficking from the ER to the Golgi. You can see them in the green right here. And the COP1 is involved into the inter-Golgi trafficking and the backward flow of vesicles from the Golgi to the ER. And this retrograde pathway from Golgi to ER is really important in retrieving the ER resident proteins that accidentally escape the Golgi but must be returned to the endoplasmic reticulum. And we talked about that in detail in one of the early parts of this topic, where the ER resident proteins have that specific signal on the C-terminus, the KDEL sequence. And the KDEL receptors retrieve the ER resident proteins from the Golgi and they move it back to the endoplasmic reticulum via these COP1 coated vesicles. So here is a transmission electron micrograph of the clathrin coated vesicles on the left and a coatomer coated vesicles on the right. This long tubular structure is likely a Golgi cisterna, but I think you can appreciate from this transmission EM that the clathrin coated vesicle have a much more regular geometric pattern on them rather than the coated vesicles. This is because the major protein of the clathrin-coated vesicle is clathrin. So clathrin is an evolutionary conserved molecule. It is made up of 6 polypeptide chains and 3 heavy chains, which are each 100 kilodaltons, shown here in pink, and 3 small chains, which are 35 kilodaltons each in molecular weight, shown in the blue. They self-assemble into this structure, this 3-armed structure, a triskelion shape. And here you can see this structure in the transmission EM. So what happens is that these triskelions come together to form a very regular geometric basket around these forming vesicles. Here is the large blue ball is the vesicle, and there are 3 triskelions which are colored red, yellow, and green, and others in blue around the vesicle. Almost looks like a cage around the vesicle, which is lined by the phospholipid bilayer or the endomembrane. So just to give you an idea that how regular these shapes are, here's a scanning EM of clathrin-coated pits and clathrin-coated vesicles. This is a cytosolic face of a fibroblast. Just to orient you, imagine yourself, you are standing inside the cell and looking up at the plasma membrane. All these clathrin-coated pits are forming towards you, so that is what you are looking at this scanning electron micrograph. There are 2 functions of this clathrin coat. One is to provide the mechanical force to form the vesicles. The second is to capture membrane receptors. So let's take a look at both of these functions. So here you have an organular membrane right here. Clathrin, other proteins in clathrin coat will begin to form or bind onto the membrane. Then they will self-assemble into that basket or cage-like structure. And it is really not known what triggers the formation of the structure, but spontaneous protein assembly to a thermodynamically minimum energy configuration and a stable state is a very normal event in the cell, with great examples of spontaneous assembly of viral proteins to make their capsids. The aggregation of the protein seems to be spontaneous. It is thought maybe when enough of these coat subunits find themselves near each other, they come together to form the basket, and it is just a thermodynamically favorable condition. It is the formation of this structure that bends the membrane right here, so it is starting to provide the mechanical forces for the formation of the vesicle. The entire invagination of the clathrin-coated vesicle depends on the assembly of that clathrin-coated pit. Finally, that vesicle pinches off, and we will talk about what regulates that in a moment. Shortly after that clathrin-coated vesicle forms, you get an uncoating because this transport vesicles likely has to fuse with another organelle. So the membrane has to be naked so that the membranes of the target organelle and this transport vesicle can interact for the fusion event. Usually the components of the clathrin coat and their proteins are recycled and used again, and we're going to talk about the mechanism in detail. So the other major function of the clathrin coat is to bind receptors. It is not clathrin itself which mediates that, but another major protein in the clathrin coat known as the adaptin or adaptin complex or the AP complex. So here is a membrane and here is a transmembrane receptor, and it is bound to some macromolecules. The dark green portion that you are seeing is the clathrin molecule. In the lighter green, right here, in the lighter green are the adaptins. So adaptins are really important components of the clathrin coat, and it is this adaptin molecule themselves that will bind to the exposed portion of the transmembrane receptor. Remember that I said that when clathrin-coated pits form on the plasma membrane, some surface receptors have the propensity to enter those clathrin-coated pits, or they are attracted to those clathrin-coated pits. What attracts them to these pits are the adaptin proteins, which are the part of this coat. So here again, you have the clathrin-coated vesicle being formed, you have the uncoating and the recycling of the clathrin coat. So the adaptins that function in the trans-Golgi network are not equivalent or are not the same adaptins that function at the plasma membrane. Remember, the clathrin-coated pit and the clathrin-coated vesicles regulate the trans-Golgi to lysosome trafficking, and they regulate receptor-mediated endocytosis. While the clathrin is the same in both of those pathways, the adaptins are different. So in this case, the cell surface clathrin-coated pits, the adaptin found here, has an affinity for proteins that have a specific sequence, a phenylalanine, then arginine, any amino acid X, and a tyrosine. So many receptors on the plasma membrane that need to be taken up by the receptor-mediated endocytosis, like the transferrin receptor or LDL receptor, have the sequence in the cytosolic portion of their cytosolic tails, and it's this sequence that interacts with the adaptin molecule. This is what attracts and holds the clathrin-coated pits. At the trans-Golgi network, the clathrin-coated buds that are responsible for forming to transport the vesicles that will move the lysosomal hydrolases to lysosomes is being shown here. Remember, the signal to move a lysosomal enzyme to a lysosome is that special modification, that mannose 6-phosphate modification, or M6P. It is the mannose 6-phosphate receptor in the trans-Golgi network that mediate that. This specific adaptins that form coats at the trans-Golgi network don't bind to the receptor sequence, but bind to specific sequence of a phosphorylated amino acid shown as this Ps. As it turns out, the mannose 6-phosphate receptors has a number of amino acid that can be phosphorylated. So the adaptin that is found in the clathrin-coated coats of the trans-Golgi network will bind specifically to the mannose 6-phosphate receptor. So what actually pinches off once the vesicle is formed? It turns out that there is a protein which is known as a dynamin, and it's a small molecular weight GTP binding protein, which means it binds to GTP and itself and has an intrinsic GTPase activity. We will talk more about the GTP binding protein in a minute, but what happens is that this dynamin will form this color on these coated vesicles as they pinch off. The intrinsic GTPase activity of dynamin cleaves or hydrolyzes the GTP to GDP. A change in conformation occurs in dynamin such that it is almost like a spring. You can picture it as a spring around the neck right here. The spring tightens up and pinches off the vesicle. So dynamin mediates that. Dynamin is that spring that wraps around the forming vesicle. You can see that very clearly here in the transmission EM. You can see the dynamin that is labeled by this electron-dense material, and you can see that how this bud is being formed here, and the pinching of the bud is facilitated by this dynamin protein. Those are the clathrin-coated vesicle. What about the codomer-coated vesicle? I mentioned that the COP1 is involved in the inter-Golgi traffic, Golgi to ER retrograde traffic, and COP2 regulates ER to Golgi trafficking. These coats are large protein complexes. There are at least 7 different proteins in these coats. We are not going to talk about each of the individual proteins. We are just going to consider the coats in general as large protein complexes. Now, in order to understand that how these coats become attached to the membrane, we have to investigate these small molecular weight GTPases because they are the important players in these codomer coats. In general, GTPases are small molecular weight monomeric or single polypeptides that can bind and hydrolyze GTP, guanosine triphosphate, to GDP or guanosine diphosphate. When GTPase is bound to GDP, the protein is inactive, and when GTPase is bound to GTP, it is active. They can toggle between the inactive and the active form based on the GDP or GTP, so they are called molecular switches. So in their inactive state, the GTPases are usually bound with the GDP or no nucleotide at all. But when they bind to the GTP, they become activated. The exchange of the GDP for GTP is facilitated by guanine exchange factor protein, or GEF, or sometimes it is called guanine nucleotide releasing protein, GNRP. Same thing, but different names. These proteins stimulates the release of the GDP, or it moves it out of the way of the GTPase so that GTPase can associate with the protein. This exchange induces a conformational change of the GTPase and it is activated and initiate various cellular proteins and pathways. Finally, the GTPase protein have an intrinsic activity which themselves can hydrolyze the GTP to GDP and therefore can shut themselves off as well. The intrinsic pathway is slow, so to speed up to this process, GTPase activating protein or GAP can enter the cycle and quickly hydrolyzes the GTP back to the GDP and therefore back to the inactive state. It can go through this cycle a number of times to regulate various functions of the cell. So here is a table that shows that actually all the coats are not just the coatomer coats, but also clathrin coats have some kind of associated GTPases. In case of COP2, an associated GTPase is known as SAR1. In the case of COP1 and the clathrin-coated vesicles, the associated GTPase is ARF and is known as the ADP ribosylation factor. So let's see how each of these functions in the formation of these coats. I should have mentioned that the function of COP1 and COP2 are very similar to clathrin. So as to assemble, they convey the mechanical force needed to form or to be able to bend the membrane to form the vesicle. So here is SAR1, which is involved in the formation of COP2 coats. SAR1 in the blue is bound to a GDP and is inactive. In its inactive form, it has a hydrophobic N-terminus, which is actually hidden inside. But at the appropriate membrane, and in the case of it is ER membrane, because we are talking about COP2, which regulates ER to Golgi trafficking, SAR1 can interact or encounter a protein known as SEC12, which is the guanine nucleotide exchange factor, or GEF, for SAR1. So when it encounters the SEC12, you get an exchange of the GDP for GTP. So now SAR1 is bound to GTP. When that happens, its hydrophobic N-terminus is exposed and it becomes membrane associated. It binds to the membrane. It is the SAR1 that recruits the components of COP2. So this is responsible for adding all the COP2 components that what we are going to see. So here it is what is shown in this slide. This is from the previous slide. So here the SAR1 in the membrane you can see is recruiting the components of the COP1 protein, so that the COP1-2 coat assembles, and this creates a mechanical force needed to form the vesicle. So here is the COP2 coated vesicle. Shortly before fusion with the target membrane, SAR1's own GTPases cleave the GTP to the GDP, so it will become inactivated. It will fall off the membrane. As a consequence of all that, the COP2 coat will come off, and that's the uncoating of the vesicle. So now you have a naked vesicle that can fuse with the target membrane. ARF function in exactly the same way, but it does for the COP1 coats and the clathrin coats. When it's bound to GDP, it is inactive. It actually has an acetyl that is buried in an inactive state. When it encounters GDP, it is inactive. When it encounters guanine nucleotide releasing factor, or GNRP, guanine exchange factor, GNF, GEF, everything is the same, shown in the green, it becomes activated because it is now bound to the GTP. GDP is exchanged for GTP. This results in a conformational change and exposes the fatty acid tail, which can be buried itself into the phospholipid bilayer. And in doing so, ARF becomes bound to the membrane. So then the ARF here shown in green will attract the components of COP1 coat or the clathrin coat, and resulting in the formation of coatomer-coated vesicles or clathrin-coated vesicles. So what is the mechanism by which vesicles form? You have these small molecular GTPases that attract these coat proteins, and it is the assembly of the coat that causes the membrane to bend the vesicles to form. So the second question we want to address, what confers the specificity in the system? How does the particular vesicle know what target organelle to bind with? So there are two major classes of proteins that regulate the specificity of the event. One is known as the snare. Other family of protein is known as the Rab family of GTPases. So let's first take a look at the snares and the Rabs. There are actually two classes of snares. One is known as a V snare for vesicle snares and become associated with the vesicle that are forming from the donor organelle. So these are the V snares. When these vesicles pinches off, those V snares are the part of the vesicle. They travel with the vesicle towards the fusing organelle. At the target organelle, you have the other class of snares known as the T snares, which stands for the target snares. The T snares interact with the V snares to help the vesicle fusion event. So this is what imparts or confers the specificity, because there are specific V snares and T snares. Depending upon where the transport vesicle is supposed to go in the cell, it will pick up specific V snare, which can only bind to the T snare on their target component. It is almost like a lock and key. So in this example, this transport vesicle that is destined for compartment B, the V snares on the transport vesicle cannot interact with the T snares on the compartment A. Likewise, the V snares on the vesicle which is destined for the compartment A cannot interact with the T snares on compartment B. That prevents the vesicle bound for compartment B from fusing with the compartment A and vice versa. So what is not shown in this slide about V snares and the T snares, and we'll look at this a little more closely in a minute, are the Rabs. It is actually the Rabs GTPases that also assist in the recognition of transport vesicle with the appropriate compartment. Now, this whole idea about these snares was proposed in the 1990s by James Rothman, and it was known as the snare hypothesis. There is enormous amount of data that supports this hypothesis experimentally. So much of that, Dr. Rothman was the co-winner of the Nobel Prize in Physiology and Medicine in 2013 for this work. It was a major important discovery in the cellular and molecular biology. So this is what it really looks like in close view. So here is your vesicle forming, and these small blue circles are the GTPases such as ARF or SAR1. The green structures are the part of the coat, COP1, COP2, or clathrin. And you can see there are some transmembrane proteins with the cargo. You can see some soluble cargo that is found associated with this vesicle. These yellow bars are the V snares. As this vesicle is forming, the specific V snares that will become associate or interact with specific T snares on the appropriate target membrane are also incorporated into the vesicle. So here is what it looks like. So now the vesicle uncoated and it's getting ready to fuse after reaching its destination, and this interaction between the V snare and the T snare that confers the specificity of this event. It actually assists in fusion too, as you will see in a minute. Now, this whole docking event of this vesicle to this target membrane is also assisted by Rab GTPases, which is not shown in the slide, but I will show you in a minute. It's important to point out that the Rab GTPases also help confer specificity. They help these transport vesicles find appropriate target membrane. I will elaborate on that in a minute. So let's move on to the last question. How do these transport vesicles, once they find their membrane, how do they fuse with the target membrane, the mechanism of fusion? So here is the V snares and the Rabs which do this. So here is your transport vesicle. It is uncoated. You have the protein here known as the VAMP. In this example, it is the V snare. Here is a Rab GTPase. Rab GTPase functions in the same way as ARF and the SAR1. They are small molecular weight GTPases. They are molecular switches. When they're bound to GTP, they're on and they're associated with the membrane. When they are bound to GDP, they are off and they are soluble in the cytosol and comes off the membrane. So the vesicle reaches the target membrane, and now it knows that this is the target membrane because the specific interaction in between the Rab and what is known as a Rab effector. You can think about this as the Rab receptor. And there is specific interaction in between the syntaxin and the SNAP25, which make up the T-snare. So this vesicle is at the correct target membrane because the Rab is bound to its receptor and the V-snare is bound to the T-snare. So I need to point out that there are a number of Rabs in the eukaryotic cell. In the mammalian cells, there are probably over 150 Rabs. They each function at different steps in the pathway. So Rab1 is involved in the ER to Golgi trafficking. Rab5 is involved in the trafficking to and from the early endosome. Rab9 is involved in trafficking to late endosome from the trans-Golgi network, and so on. Here is how Rabs confer their specificity, as you can see some of them here. Depending on where the vesicle is budding will dictate which Rab interacts with what particular forming vesicle. Of course, the target membrane will have the appropriate Rab receptor as well. So the Rab binds to its receptor, and there's a weak interaction between the T-snare and the V-snare, and immediately followed by a very strong interaction between the V-snare and the T-snare. In fact, what happens is that T-snare and the V-snare become intertwined. So in this process, it is pulling down the vesicle towards the membrane. So here is a ribbon diagram, which is a little bit larger so that you can see that they intertwine. And it is actually the association of the V-snare and the T-snare and this twisting of the V-snare with the T-snare that bring this uncoated vesicle right up to the target membrane. And then you have a spontaneous fusion event. Please take a look at the additional supplementary videos for a clear understanding of the event. So you can see that there is this vesicle has fused with the target membrane, and the entire V-snare and T-snare complex is also in that membrane. Now we are going to talk about the fate of these snares, which will be using these proteins to get recycled. So you need to have an opportunity to recycle the V-snare and the T-snares, and that is carried out by a protein complex that is made up of proteins known as NSF, which is a homohexameric protein in the dark green. Another protein attached to it is the alpha SNAP. What NSF and alpha SNAPs do is using the energy stored in the ATP by harnessing the energy of ATP hydrolysis, the snare complexes are disassembled, and the VAMP, which is the V-snare, is recycled back to the donor membrane, and that completes the mechanism of vesicle membrane fusion and recycling of their product. Now you will be able to appreciate the cover picture of the chapter 14, which shows a clathrin-coated vesicle formation, how these three-legged clathrin triskelions are being assembled, and it's pinching off the membrane, underlying membrane, and then it assembles into a clathrin-coated vesicles for the final scission of the membrane and yields a coated vesicle. And this is also the end of module 2.1.